So I want to tell you about the regulation of Clathrin-mediated endocytosis. And it's important because, as you know, the plasma membrane is a physical barrier that separates a very different milieu outside the cell from inside the cell. But it's also a sophisticated communication device. So cells talk to each other and they interact with their environment through the plasma membrane. And so endocytosis, is that not? It's already frozen. Let me try that. There you go. Endocytosis, which is the uptake of macromolecules from the cell surface, controls the composition of the cell and therefore how the cell can take up and nutrients from its environment. Things like glucose and insulin metabolism, signaling from the cell surface. And so endocytosis must be tightly regulated. The major endocytic pathway in mammalian cells is Clathrin-mediated endocytosis, which is a multi-step process that starts with initiation. This is when the adapter proteins bind to the plasma membrane and recruit the major coat protein clathrin. The continued assembly of adapters in clathrin helps to recruit cargo molecules into a growing and invaginating coated pit. These coated pits mature through the aid of numerous endocytic accessory proteins, and then finally the GTPase dynamin assembles into collar-like structures at the necks of coated pits to pinch off coated vesicles carrying their concentrated cargo into the cell. Finally, an uncoating reaction disassembles all of this apparatus for multiple rounds of endocytosis. The dynamin GTPase is a paradigmatic fission catalyst. And here is one model of how dynamin catalyzes fission based on about 20 years of biochemistry from my lab. Dynamin assembles first using mechanochemical action into these collar-like structures, helping to squeeze the necks of deeply invaginated coated pits. Multiple rounds of GTP binding and hydrolysis, which drives dynamin disassembly, helps to optimize the size of the collar into just the right dimensions to facilitate membrane fission. A key high-energy transition state conformation of dynamin causes wedging of the membrane-associated pH domains into the lipid bilayer, destabilizing that lipid bilayer and therefore catalyzing fission. The collar itself can be a barrier to fission, and so GTP hydrolysis again helps to loosen the collar, allowing the reaction to be completed. One of my favorite quotes is this from Daniel Bernstein, that the greatest obstacle to discovery is the illusion of knowledge. So we think of dynamin as the paradigmatic fission machine, but indeed, dynamin appeared evolutionarily much after Clathrin-mediated endocytosis. So you see the appearance of clathrin, adapters, and many of the endocytic accessory proteins since the very first eukaryotic ancestor cells. But dynamin, you see, is completely absent throughout all of these early stages of evolution until you get to multicellularity. So clearly, fission of clathrin and clathrin-mediated endocytosis were occurring for eons before the advent evolutionarily of dynamin. So it can't just be a fission machine. So work in my lab over the past about 15 years has revealed that, in fact, dynamin can regulate these early stages of endocytosis in its unassembled state. And then assembles into these collar-like structures to catalyze membrane fission. We believe the transition from these early to late stages of dynamin function is mediated by an assembly switch. Although dynamin is a GTPase, it is not controlled like other GTPases by a nucleotide binding switch, but we think instead by this assembly switch, and that factors work to control dynamin assembly and disassembly. So this is a great model. Another favorite quote of mine, all models are wrong, but some are useful. And we have been trying to test aspects of this model. So another sort of myth about dynamin is that invertebrates, which express 3 dynamin isoforms, it's believed that dynamin-1 is a neuron-specific isoform and dynamin-2 is ubiquitously expressed. But if you look carefully at the scales of this microRNA data, you see that in fact, and adjust them accordingly, you see that in fact dynamin-1 is messenger RNA levels are present at equal or higher levels than dynamin-2 messenger RNA levels in all tissues. It's just hugely overexpressed at the synapse. So this made us ask, what is dynamin-1 doing outside of neurons, and how is dynamin regulated in these non-neuronal cells? Some years ago, we did some biochemical studies to look at the differences in biochemical properties between dynamin-1 and 2, and to summarize some very nice work by Yawen Liu and Sylvia Newman in my lab. We showed that dynamin-1 is a potent curvature sensor, that is a generator, that is given a flat membrane or liposome, it can bind to that and squeeze that liposome into a narrow diameter. It's ideally suited for this very rapid endocytosis and recycling that occurs at the synapse, sometimes even in the absence of clathrin, and dynamin functions in this entire synaptic vesicle recycling pathway. In contrast, dynamin-2, the more ubiquitously expressed, or previously thought to be ubiquitously expressed isoform, is a curvature sensor. It cannot, on its own, squeeze membrane tubules. Instead, however, if it's given a narrow tubule, it can cut them. And so dynamin-2 won't assemble onto that narrow neck until the neck is created by other endocytic accessory proteins. And in fact, in vitro, dynamin-2 works synergistically with bar domain proteins to catalyze membrane fission. So Clathrin-mediated endocytosis in non-neuronal cells requires dynamin-2, but not dynamin-1. So here, as I showed you in these, or I told you in these RPE cells, they express roughly equal levels of dynamin-1 and dynamin-2, but only siRNA knockdown of dynamin-2 inhibits Clathrin-mediated endocytosis in these cells. Knockdown of dynamin-1 has no effect. Some years ago, we discovered when studying a mutant of Clathrin-mediated endocytosis, I won't go into the details of what the mutant is for the sake of time, but a mutation that severely impaired the maturation of Clathrin-coated pits and maturation of Clathrin-coated pits and vesicles. Despite this defect in maturation, Clathrin-mediated endocytosis in these cells occurred as robustly as in wild-type cells. And what we discovered is a compensatory mechanism whereby these cells activate dynamin-1. So there's no changes in the expression levels of dynamin, but you can see that now these cells require both dynamin-1 and dynamin-2 for Clathrin-mediated endocytosis. The two isoforms still have non-redundant functions, as both are required for endocytosis in these mutant cells. So how is dynamin-1 being activated? We know a lot about how dynamin-1 is activated at the synapse in response to an action potential in for synaptic vesicle recycling. It turns out that in the resting synapse, dynamin-1 is maintained in a phosphorylated inactive state by a constitutively active kinase, GSK3 beta. Upon activation of the synapse, calcium rushes in, activates this calcium-dependent phosphatase calcineurin, which dephosphorylates dynamin-1 to activate it. This constitutively active kinase is itself regulated by another kinase, AKT, or phosphokinase B, which phosphorylates GSK3 beta to inactivate it. So we wondered whether these mechanisms of regulation of dynamin might be active in the cells that we were studying. And indeed, that turned out to be the case. So first, if you look at control cells and the wild type cells in black, when you inhibit GSK3 beta and prevent the phosphorylation of dynamin 1, Clathrin-mediated endocytosis is accelerated in normal cells, but unaffected in the mutant cells because the dynamin 1 is already activated. In contrast, if you inhibit AKT, it has no effect in the control cells because dynamin 1 is already inactive, but inhibiting AKT, which inhibits GSK3 beta, so that now the GSK3 beta is active, inhibits endocytosis in the mutant cells. So these two kinases are operating to regulate dynamin 1 function in non-neuronal cells. And this is true in the mutant cells, but it's also true in normal RPE cells. So now we're just taking normal parental RPE cells. And as I showed you before, I'll just walk you through this complex slide, a lot of data on this slide. As I showed you before, in these normal cells, if you knock down dynamin 1, you have no effect on Clathrin-mediated endocytosis. Here we're looking at coated pit dynamics using total internal reflection fluorescence microscopy, where we can measure the distribution, the frequency, the distribution of lifetimes of all the coated pits budding off in these cells. And so again, you can see that knockdown of dynamin 1 has no effect on the lifetime distribution of coated pits in these cells. If you acutely inhibit GSK3 beta, you see this increased rate of Clathrin-mediated endocytosis and a marked change in the lifetime distribution of coated pits in these cells. Instead of this Rayleigh-type distribution that reflects tightly regulated process, you now have a near exponential distribution reflective of a more rapid dysregulated endocytosis. This GSK3 beta effect, importantly, is completely on endocytosis, is completely dependent on dynamin 1. So in the absence of dynamin 1 in the dynamin 1 knockout cells, you no longer see any effect of GSK3 beta inhibition. However, it's still dependent on dynamin 2. So as I told you, dynamin 2 knockdown inhibits endocytosis in these cells, but that residual endocytosis is still stimulated by addition of GSK3 beta. So not only is dynamin 1 activation increasing the rate of maturation and decreasing the lifetime of Clathrin-coated pits, amazingly, it's also increasing the very, very first step, the rate of initiation. So here again, when we treat these cells with GSK3 beta, we see a 2 to 3-fold increase in the rate of formation of new coated pits. And this, again, is completely dependent on the expression of dynamin 1 in these cells. So dynamin 1 is ubiquitously expressed, and when it's activated, it accelerates these very earliest stages of Clathrin-mediated endocytosis, coated pit initiation and maturation. So I mentioned at the beginning that endocytosis controls signaling. And now I'm showing you that signaling can also feed back and regulate endocytosis through this GSK3 beta AKT kinase cascade. So AKT you might have heard of, as it's an important oncogenic kinase in many cancer cells. So we wondered whether these effects of AKT might be operating in cancer cells pathologically. And this turns out potentially to be relevant because if you look at expression levels of dynamin 1 in 50 normal lung epithelial cells versus a panel of 108 non-small cell lung cancer lines, you can see that dynamin 1 in general is upregulated in cancer cell lines over the normal cell lines, whereas expression levels of dynamin 2 are unchanged. And indeed, if you look at survival curves of patients that express low versus high dynamin 1, lung cancer patients, you can see that there's high dynamin 1 expression is a poor prognostic marker for survival, especially if you're a smoker. So we think this is physiologically relevant. So we looked in H1299 cells, a non-small lung cancer line known to have active AKT, and you can see in these cells already the hint that endocytosis is partially dependent on dynamin 1, and of course still dependent on dynamin 2. Interestingly, endocytosis in these cells is highly sensitive to inhibition of AKT. So if we inhibit AKT, which no longer inhibits GSK3 beta, we inactivate dynamin 1 in these cells. So GSK3 beta in these non-small cell lung cancer cells, like in the normal RPE cells, inhibition further increases the rate of coated pit initiation, further decreases the lifetime of coated pits, and alters the lifetime distribution to create more quasi-exponential decay. And all of these effects of GSK3 beta inhibition are dependent on dynamin 1 expression. So if we knock out dynamin 1 in these lung cancer lines using CRISPR-Cas9, you see that all of these effects of GSK3 beta are abrogated. Interestingly, it doesn't take much dynamin 1 in cells to have these effects, so that here we used genome editing to tag endogenously dynamin 2 with the M-cherry and dynamin 1 with EGFP. And you can see in these, we have a heterozygous dynamin 2 M-cherry clone that we've studied, and likewise a heterozygous dynamin 2 EGFP clone. In these H1299 non-small lung cancer cell lines, we can barely detect dynamin 1 by Western blotting. We have to concentrate the dynamin 1 first by pulling it down with a SH3 domain amphiphysin, and then we detect dynamin 1. So in your cells, if you say there's no dynamin 1, it just may not be detectable yet. As I've showed you, it still can be activated and profoundly affect the rates of endocytosis in these cells. So dynamin 2 in these cells is recruited at the late stages of endocytosis to catalyze these fission reactions, as I've shown you before. Dynamin 1 has detected at much lower, almost undetectable levels to these coated pits, and we see no evidence of this late burst. It's recruited uniformly throughout the lifetime of a coated pit. Well, there's another step in the early endocytic pathway that we've now linked to the activation of dynamin 1, and that's the accumulation of a special population of endosomes located at the extreme cell periphery that are called APPL1 endosomes or signaling endosomes. The scaffold protein APPL1 recruits, it turns out, both AKT and GSK3 beta. So these early endosome populations, which are the sites of very rapid recycling of receptors back to the cell surface, are also the sites at which AKT and GSK3 beta interactions and phosphorylations occur. And so you get the sense that this APPL1 endosome and its accumulation might provide a feedback to increase and maintain the activity of dynamin 1. And indeed, dynamin 1 is selectively required for the peripheral accumulation of these APPL1 endosomes. Again, back in normal RPE cells, you can see in the normal RPE cells, this is now an inverted fluorescence, the distribution of APPL1 endosomes at the periphery of these cells detected by turf microscopy. If you knock out dynamin 2, you have no effect on this distribution. If you constitutively knock out dynamin 3, you have no effect on this distribution. Only if you knock out dynamin 1 in these cells do these APPL1 endosomes now disperse. So this indeed creates a positive feedback loop for regulating Clathrin-mediated endocytosis, in particular in cancer cells. So we find that in cancer cells, they have these alterations in Clathrin-mediated endocytosis that activates dynamin 1 to increase the distribution and accumulation of these APPL1 scaffolding endosomes, which in turn increases activation of AKT, which changes the endocytosis and continues to activate dynamin 1. And this is a true feedback loop because the increased rates of endocytosis we see in these cells are sensitive to AKT inhibition, and the activation of AKT in these cells requires dynamin 1, as it's not seen in the dynamin-1 knockout cells. So this positive feedback loop can be activated in cancer cells to alter endocytosis. So what we think is happening is cancer cells are activating neuronal pathways of endocytosis, which are pathways that teleologically are designed for cell-cell signaling and maintenance and sustaining that cell-cell signaling at the synapse in the case of rapid synaptic vesicle recycling. And we believe that this same rapid recycling pathway, endocytosis and rapid recycling of growth factor receptors like EGF receptors, c-Met, et cetera, are enhancing and sustaining their signaling, and in so doing, enhancing the proliferative and migratory and metastatic activities of cancer cells. So to summarize then, what I've told you is that dynamin-1 also regulates these early stages of Clathrin-mediated endocytosis in non-neuronal cells, that there's reciprocal crosstalk between signaling and endocytosis. And I think this is an important concept, that the cargo, these active signaling receptors, aren't just passively moving into coated pits and being taken up. They can actually change the dynamics of that pathway and affect their own rapid recycling through these APPL1 signaling early endosomes, the proliferation and accumulation of these APPL1 signaling endosomes to control their own fate. So this dynamin isoform switch that occurs in some lung cancer cells can dysregulate endocytosis and these early stages of endocytic trafficking to the benefit to alter the signaling properties of these cancer cells. And I like to think of this as adaptive Clathrin-mediated endocytosis that functions as cancer cells undergo their own evolution to alter signaling, enhance proliferation, survival, and metastasis. So I'll close almost on time. Look at that. By thanking the people in my group that have done this work. Primarily, this work was initiated by Carlos Reyes and Ping-Hung Chen in the lab. Many of the CRISPR knockout lines and knock-in lines have been generated and studied by Praveen Srinivasan, and Marcel Metlin is our guru of turf microscopy, and I haven't been able to talk about the work done by others in the lab. So thank you very much for your attention.